History, okay. How about the product details? Can you share with me, Ika?
Next, we move to the product details. Iprex prepared strains contain the active ingredients of epoetin alpha. So what is the epoetin alpha? Epoetin alpha is a protein that stimulates bone marrow to produce more red blood cells. Red blood cells are responsible for carrying oxygen to all parts of our body. A decrease in the number of blood cells can cause anemia. Here we have active ingredient, inactive ingredient, and also available dosage. So epoetin alpha is the active ingredient of this product. For inactive ingredients, we have the sodium phosphate dehydrate, glycine and water for injections, polysorbate 80, sodium chloride, and also sodium dehydrogen phosphate dehydrate. We have 4 available dosage, which is 2000 IU per 0.5 ml, 4000 IU per 0.5 ml, 10,000 IU per ml, and also 40,000 IU per ml.
So, Ika, how epoetin alpha works in our body?
Oh, sure, I will tell you how. Then we move to the biological concept. At this part, it will tell how the biopharmaceutical product works and what is their mechanism of action. So basically, the MOA of erythropoietin is that they will regulate the production of red blood cells by stimulating the division and differentiation of committed erythroid progenitor cells in the bone marrow. It works by causing the bone marrow, which is soft tissue inside the bones where blood is made, to make or produce more red blood cells. For example, a patient who has anemia has lower oxygen levels in blood. It will go to the kidney, the place where erythropoietin releases. Then the erythropoietin will stimulate red bone marrow to enhance erythropoiesis, which increases in red blood cells count. By increasing in red blood cells will increasing oxygen level that has the ability to carry blood in all parts of the body. So that patient will have normal blood levels. The manufacturing process of epoetin alpha using recombinant DNA technology involves several steps. First, gene isolation. The gene that codes for human erythropoietin is isolated from human DNA. This gene contains the instructions for producing the EPO protein. Next, the gene cloning. The isolated EPO gene is inserted into a plasmid vector, a small circular DNA molecule. This creates a recombinant plasmid containing the EPO gene. Host cell selection. The recombinant plasmid is introduced into host cells, typically Chinese hamster ovary cells. CHO cells are often used because they can efficiently produce complex human proteins. For cell cultivation, the transformed CHO cells are cultivated in bioreactors, providing them with a controlled environment for growth. These cells multiply and express the EPO protein. For protein expression, the EPO gene in the recombinant CHO cells is transcribed into messenger RNA, which is mRNA, which is then translated by the cellular machinery to produce the EPO protein. For protein secretion, the EPO protein is synthesized and secreted into the culture medium by the CHO cells. For harvesting, the culture medium containing the secretion EPO protein is collected from the bioreactors. For purification, the harvested culture medium undergoes purification processes to isolate and concentrate the EPO protein. These processes involve various chromatography steps to remove impurities and unwanted substance. For formulation, the purified EPO protein is formulated into the desired dosage form, usually an injection solution. This formulation ensures stability and appropriate delivery of the drug. For quality control, rigorous quality control tests are performed at different stages of the manufacturing process to ensure the final product meets safety, efficacy, and quality standards. These tests include checks for protein identified, purity, potency, and absence of contaminants. For fill and finish, the final purified EPO solution is filled into vials or other appropriate containers. The containers are sealed and labeled for distribution. For packaging and distribution, the packaged EPO vials are ready for distribution to healthcare providers and patients. So in conclusion, can conclude that currently available hematopoietic growth factors can improve the production of a major blood cells groups, which is red blood cells, white blood cells, and platelets. The availability of these growth factors has had a major impact on helps in the treatment of conditions like anemia, neutropenia, and thrombocytopenia. As with any other therapy, the use of these growth factors must be assessed with regards to their proven clinical efficacy, side effect profile, and cost containment measures. Last but not least, a greater understanding of these elements might eventually result in therapies that are more focused and efficient.